Hi, welcome to another lecture about general embryology part 3. I hope you have a nice day. I don't feel like putting myself on camera today, so you can have a nice view on presentation, and let's start. The first part we will deal with extra embryonic structures, the chorion, placenta, umbilical cord. This part is very important for you. And the second part of the lecture, I would like to summarize the development in the last weeks. So let's summarize such an overall view of the fetus and the newborn. Let's start with the first part, the development of amniotic sac, like the fetal membranes, the placenta, and umbilical cord. You probably know these pictures from previous lectures. This first picture is the 2-cell stage embryo right here. This is after 30 hours after fertilization. Here we have 2 blastomeres, one and a second one. These are called daughter cells, which already undergo mitosis. So after one cleavage we have 2 daughter cells called blastomeres, and these cells are totipotent. And what does it mean, totipotent? It means that each cell, each blastomere, can give rise to whole embryo. So each cell can develop into whole embryo. And after mitosis, we have 4-cell stage, so we have 4 blastomeres in about 40 hours after fertilization. And also it's the same as for the 2-cell stage, so also these blastomeres are totipotent. But when it becomes a morula, so now we have a morula, here we have 16 blastomeres in morula after another mitosis. The cells begin to differentiate more and transform into the inner cell mass, or so-called embryoblast. I'm just going to put E here, like for embryoblast. Which is eventually going to differentiate into embryo. That's why it's called embryoblast, the inner cell mass. And we have outer cell mass, or trophoblast, which is going to differentiate into those things that are extra embryonic, so everything else. Not embryo, so the placenta, umbilical cord, and the chorion, and these stuffs. So today we're going to focus on outer cell mass and the trophoblast and what is going on there. Here you have nice pictures, so you can see how it really looks like. This is a nice morula over here, very nice. And this is the 2-cell stage. You can see this red layer that is around blastomeres, and you probably know it's called zona pellucida. And in a process called hatching, we're going to get late blastocyst, which doesn't have zona pellucida around. So this process, when it loses the zona pellucida, it's called hatching. So this is the late blastocyst. You can probably recognize it. We have a nice inner cell mass, embryoblast, this one in blue, and the green one represents the outer cell mass, the trophoblast. So once again, we're going to focus on this extra embryonic layer right here. And here we have a nice blastocyst cavity in the inside. And what is happening is that the blastocyst begins to be implanted. It needs to be implanted in the uterus right here. So here we have a uterus, probably somewhere here, but what we can see is the uterine epithelium, which you can probably recognize right now. We are going to learn more about uterus in the female system in the second semester. But now you can tell that it's a simple columnar epithelium. Simple columnar epithelial cells are right here. So this is the normal epithelium of uterus, and together with layer that is under epithelium, this is the mucosa. So embryo is going to be implanted in the mucosa right here. But mucosa needs to change a little bit. Yeah, it needs to be changed. And it's undergoing a so-called decidual transformation. The mucosa of the uterus is undergoing decidual transformation because a mucosa of the uterus normally contains fibroblasts. Here, fibroblasts, which are normal cells that you already know. Fibroblasts producing collagen and stuff in a normal loose connective tissue and also in dense connective tissue. So normal cells also in the uterus, and they change into decidual cells. Decidual cells. And these cells are very different than fibroblasts. They are no longer thin, they are very huge. They are pink, they contain lipids and they contain glycogen. So they are ready to nourish the embryo. Yeah? So fibroblasts change in the decidual cells, and this is called decidual transformation. They're going to ask you for sure during exam what is the decidual transformation when you get these pictures over here. So you can see right now the proliferation of trophoblast. You know, the trophoblast is going to differentiate into two types of cells, but these trophoblastic cells in general are proliferating and making these long protrusions to get to the mucosa of the uterus. So this is still the first week. Yeah, first week. This is happening first week after fertilization. And now we are in the second week. We have 9 days right here. And this is starting to be very interesting right now. So we have a bilaminar embryo here made of epiblast and hypoblast. That's why it's called bilaminar embryo. But let's focus on the trophoblast. So the trophoblast is differentiated now into two layers and is differentiated into syncytial trophoblast. This very dark green layer over here, syncytial trophoblast, it's a syncytium of cells. You already know some syncytium, like a muscle fiber or osteoclast. So once again, this is also syncytium. And cytotrophoblast over here under syncytium, this light green color, these are just normal cells that are dividing and making syncytial trophoblast. So cytotrophoblast develops into syncytial trophoblast gradually. In the syncytial trophoblast, lacunae begin to appear. Lacunae, these spaces over here, which are empty right now, or not so empty, because there is no empty space over here, just filled with air, not happening here. They are filled with just fluid. Yeah, there is just some fluid, interstitial fluid, and also secretions of glands are here in this fluid. And so the embryo nutrition is histotrophic. The nutrition is histotrophic. I'm going to write it like this, but you get the point. It's histotrophic because embryo is nourished only by the glands and the decidual cells that are around in the uterus, not by blood. Yeah, so it's the nourishment is only by the decidual cells and the glands in the uterus. As the embryo grows, the surrounding tissues are no longer sufficient, and embryo begins to suffer from hypoxia. Yeah? So the supply of oxygen is becoming very important. And what's happening? You can see these vessels over here, maternal vessels. Yeah, so we need to somehow get to this blood over here, because this is no longer sufficient for us. Just the fluid and the glands, No, we want to grow big, so we need some blood over here. So what's going to happen is that these maternal vessels will respond and they start to enlarge. And these capillaries that are in the uterus, they are normally in the uterus, in the wall, in the mucosa, they acquire the character of sinusoids. You're going to learn more about sinusoids in the next lectures. Sinusoids are just like big white capillaries which are ready for transfer metabolites. Yeah, so for the exchange of metabolites. Yeah, they are very big and loose. So normal capillaries transform into sinusoids here. And syncytial trophoblast starts to go and start to disrupt to the capillaries, the wall of capillaries. So we're going to see it in the next stage. Here also, this is just a picture, normal histological picture. Hematoxylin eosin. No, probably we have some other staining over here because I can see the blue over here, so it's very complicated in this picture, but it isn't that important. What's important is that we can see the lacunae over here. Yeah, so the space is filled just with fluid. Here probably they are just empty because of the preparation, but this is the lacunar stage of the embryo, lacunar stage. Lacunae is filled with fluid. Also, we can see it with higher magnification over here. So, as I said, gradually the syncytial trophoblast disrupts the capillary wall, and maternal blood enters the lacunae. Thus, from histotrophic stage, it begins the hemotrophic stage. Now we have hemotrophic stage. So the feeding of embryo from the mother's blood, actually, this is what it means. And this will continue. And as you can see, oxygen, there are no vessels here, so oxygen can diffuse from the mother's blood and nourishes the trophoblast mostly. Yeah, so here is very good diffusion. And we have also extra embryonic mesoderm here around. So also extra embryonic mesoderm is pretty well nourished. You know that extra embryonic mesoderm is divided into splanchnopleural and splanchnopleuric mesoderm, which is over here, this layer, and somatopleuric mesoderm, which is close to the trophoblast over here. And between these two layers, an extra embryonic coelom is developing. So some spaces are made here and coelom is developing. And the splanchnopleuric mesoderm is very close to the exocoelomic cavity or primitive yolk sac. Yolk sac is maybe a better name for it. This is too complicated, exocoelomic cavity. Yeah, you can really confuse these two together, so let's just call it yolk sac for our purposes. But look at the distance of the diffusion. Yeah, the distance to this bilaminar embryo is very large. So the diffusion isn't sufficient enough for the embryo. So what's going to happen? Embryo starts to lag behind in growth. Yeah, and somatopleura, the part of extraembryonic mesoderm, and trophoblast begin to expand because they are very well nourished by a process of diffusion, and other structures lag behind in growth. We're going to see that. Just now imagine what's going on here. The trophoblast is very well nourished, but the embryo, no, that's just too long way for the blood to go here by diffusion. Here we have once again a nice picture, histological picture. What we can see here are these lacunae that are filled with blood. And of course there is a maternal blood over here, yeah, which is like nourishing everything around. And once again the next lacune over here. Here probably we are able to see decidual cells right here. So compare this cell to a fibroblast. Yeah, it's very different. It's bigger cell, also here, big cell, eosinophilic, filled with lipids and glycogen and other stuff. Yeah, but really the decidual cells themselves lost their importance for nutrition already during this period. Yeah, because we have maternal blood, so decidual cells are just sitting there, but they're not so very important as they were before, but they persist until the end of pregnancy. Here we have a cytotrophoblast over here. Just a layer of that's just a layer of cytotrophoblast, and here we have a syncytotrophoblast containing lacunae. But it's just for the visualizations. We really don't have slides that are this complicated. Okay, so and now you can see it. Yeah, here's the third week, the day 20 days after fertilization, and now we can see the disproportion as the trophoblast has grown and the trilaminar embryo, right here we have trilaminar embryo, is already lagging behind. You can see how small is the embryo. Really, it's because it doesn't have enough nutrition over here, but it's very normal state. It's not the pathology, it's normal state like this. It won't take long, actually. You can see it right here. This is the connecting stalk, over here, connecting stalk. And also you can see it here in extraembryonic mesoderm, right here. The vessels start to form. Blood vessels begin to form here. And the blood begins to flow very irregularly in them. So it's just splashing from one side to another side. And after the formation of the heart and vascular system, blood starts to flow regularly. So now here we have some new blood vessels over here. And we're forming these vessels to nourish the embryo itself. And what we can see also in this picture is fully developed chorion right here. It's fully developed chorion. So this is the chorion right here. Yeah, I'm going to draw it like this in the circle. So this is the chorion over here. The chorion consists of trophoblast. I'm going to probably write it down. So we have chorion that consists of trophoblast, which is of course divided into syncytotrophoblast and cytotrophoblast. Yeah, and also it contains an extra embryonic mesoderm, specifically this layer over here, which is called, you probably know that, and it's a somatopleura. But remember, we are in the extra embryonic tissues. Yeah, so somatopleura of extra embryonic mesoderm. So really, chorion have two parts, I guess. So trophoblast and extra embryonic somatopleuric mesoderm. So the embryo was completely implanted, and the pathway where it entered the endometrium healed completely. So now we can see a really nice layer of simple columnar epithelium of the uterus. So we are no longer can see where it was implanted. Right here, it's completely healed. Because, you know, at the end of the second week, a fibrin clot was seen here, a fibrin clot. But now, here we have just continuous epithelium. So, we can see the lacunes over here. Still, here we have lacunes, but you already know them. And more nutrients can get to the chorion that is more inside on this side. So the supply of chorion on this side over here, I'm going to draw it like this, there are many colors right here. So this not so many because it's only blue, but I think many lines are over here. So this part is supplied more than other parts of the chorion because it's a part that is deeper in the mucosa. And here we have the same situation as before. When it's something deeper and more close to supply, it's going to be bigger. Yeah. While the other side really is just surrounded, this other side over here is just surrounded by a thin layer of decidua and the cells that are here. So this side, the other side over here, I'm going to label it with green, isn't supplied as well. So this pole doesn't grow so fast. The chorion on this side, the blue part here, that grows more and forms villi. We can really find villi over here, which I'm going to describe later, is called chorion frondosum. So this part over here is chorion frondosum, or the viscous, it contains villi, and really what I wanted to get to, and the chorion that's on the opposite pole right here, doesn't contain villi that much. Maybe we can see some villi over here, another one over here, but it's more smooth than other chorion over here, which is supplied better. And it's called chorion leve, or smooth chorion, because it's really smooth. So, and we have also decidua around these layers, and also decidua, there isn't only one decidua, there are many types of decidua, and we distinguish the decidua basalis, or the basal decidua, which is very close to the chorion frondosum. So the layer here that is close and it belongs to the mother, it's the part of the uterus, is decidua basalis in Latin, or the basal decidua. So if there will be question like, what's the basal decidua? You just say that, okay, it's the part of decidua that is close to chorion frondosum. It's part of the mother's uterus right here that is close to this part of the fetus. We further distinguish decidual capsularis, or the decidual capsule, which is on the other side, where we can find the chorion, the smooth chorion over here. So this decidua over here, it's called capsular decidua. And the basal decidua and capsular decidua are connected with decidua that is here, that is called marginal decidua. on both sides. So I hope you understand. We have the decidua basalis or basal one, the marginal one, and the capsular one, which is close to the smooth chorion over here. This chorion is smooth because it does not develop so quickly. Yeah, so it's lagging behind the chorion frondosum because it hasn't got so much nutrients here. So just remember, capsule decidua is close to smooth chorion over here. Basal decidua is close to frondosum, the chorion frondosum over here. Chorion villi, actually, chorion villi are very important formations. In this case over here, I'm going to delete everything. So here we have once again chorion frondosum, and these are the villi over here. This is the villus, like this over here. I'm going to dissect it right here. So these are the villi over here, and these are very important structures. These are the tertiary villi. Tertiary villi because they contain vessels. I'm going to explain it in a moment. These villi are important for the nutrition and construction of placenta. So, here we can see the development of chorionic villi over here. At first, they are only made of trophoblast over here, so it's type A. On the surface there is a syncytium here on the surface, and inside there we have a cytotrophoblast over here. And this villus is called primary villus right here. This stage really immediately changes into secondary villus over here, which is already filled with somatopleura, or so we can call it mesoderm core, but you know that it's filled with somatopleura. I'm going to write it down. Pleura. So here we have extra embryonic mesoderm inside, and this type of villus is called secondary villus. So we have first primary villus, then we have secondary villus. And when blood vessels grow into the mesoderm over here, because it's going to happen, really, blood vessels will be created here in the mesoderm, it turns into tertiary villus. Tertiary villus. All around the villi, here, there is maternal blood everywhere around. So here we have maternal blood. This maternal blood, you know, it's in the lacunae, yeah? And these lacunae will fuse in a form that these lacunae will create a space, the whole space, that is called intervillus space. Intervillus because it's between the villi, so it's very meaningful. Yeah, so intervillus space around these villi over here, and here we have maternal blood around villi and fetal blood on the inside of the villi. So there is no way that blood of the fetus will mix with maternal blood. There is some kind of barrier right here. Yeah, the fetal and maternal blood are not in a direct contact. Yeah, there is this certain barrier which will not let everything pass through. Yeah, only some substances will pass through, like blood gases and nutrients and various hormones and other stuff we are going to learn, and I'm going to tell you more about that maybe in next lectures, and you're going to learn about that in physiology and other subjects. So, what's happening really here? In the case of secondary villus, when we have only secondary villi, those nutrients only need to be diffused. So we don't have vessels, they need to be diffused. But in the case of tertiary villi, they pass into the vessels and are transported further. In the tertiary villus, we see the maturation of the villus as the cytotrophoblast, that is over here, gradually decreases, really to facilitate the transfer of nutrients. So we have less and less cytotrophoblast. We have a nice layer of syncytiotrophoblastic here, but there are less and less cytotrophoblastic cells over here. And so this is well mature, well developed tertiary villus over here. And these stages are very important. You should know everything that you can find in primary villus, in secondary. There are many questions in tests related to this, so just remember what does it mean. And in placenta, the slide of placenta that we have in our compartment, you can find only tertiary villi, because these tertiary villi are in a mature placenta. Yeah, so we can find tertiary villi, only tertiary villi in mature placenta. So here we can see some vessels in the inside, and the arteries are really there is some difference in fetal circulation, because the arteries are generally the vessels that carry blood from the heart and contain oxygenated blood. In normal adults and kids and normal human beings, arteries that go from the heart are called arteries. Yet vessels that go from heart are called arteries, and they contain oxygenated blood. And the veins are the vessels that lead to the heart and have non-oxygenated blood. Yeah, that's just normal. But the situation is different in fetus, because in the fetus, however, it's opposite, because the fetus receives nutrients from the mother. So the vessels that lead to the heart, contain oxygenated blood. So basically, the veins contain oxygenated blood, if you know what I mean, yeah? So probably these vessels that I'm encircling, these red ones, the red probably means oxygenated blood over here. These vessels are heading to the heart of the fetus, and these vessels are veins. Yeah. Also, they are veins. They contain oxygenated blood, but we call them veins because they're heading to the heart. So it's opposite than in normal stage. And how do we form these vessels? I don't know if I have I don't have a picture on that, but you're going to learn more about formation vessels in the cardiovascular system. But now you should know we have two basic processes, namely, first process is called vasculogenesis. This means the formation of new blood vessels. So I'm going to just write down the new blood vessels over here. This is vasculogenesis. So we have some mesoderm, and then boom, we have vessel. Yeah, that's vasculogenesis. And the second process is called angiogenesis. And this means the sprouting of already formed vessels. So we created the vessel, and it's going to branch, and this branching, like the creation of new vessels from already existing vessel, is called angiogenesis. Yeah, so just know these two processes. They are very important. Also, many questions related to these processes. And don't forget about the space that is around. It's called intervillus space. So now we have a nice picture from placenta. And what do we have here? This is the section of villus And you probably know this is normal mature placenta. So this villus right here is tertiary villus. We can see vessels inside. So of course it's tertiary villus. And also around vessels we have well differentiated mesoderm, and now it's in the form of loose connective tissue. So normal placenta have villi that are filled with loose connective tissue. This loose connective tissue differentiate from extraembryonic mesoderm, of somatopleura right here. So vessels and loose connective tissue. There is a syncytial trophoblast covering the villus. So here we have a nice layer of syncytial trophoblast over here. And sometimes we can see right under syncytial trophoblast there are cells, separate cells of cytotrophoblast. And I can see really nice cell over here. This is the cell of cytotrophoblast. And also one cell over here. But we are going to focus more on this slide during next semester, so don't worry, we will get to this. This is just like brief describing really, and there are many more things that we can see here, but remember there is a syncytial trophoblast on the top, there are some cells of cytotrophoblast, there are vessels, and there is loose connective tissue around, so we call this villi. This is just a section and we call it a tertiary villi over here. Also we have some villi also over here. This is another villus and this is another one right here. So remember this, these cells are in the villi, so they belong to fetus. These are the fetal cells, vessels, fetal vessels, these vessels. And around villus we have intervillus space, because it's between the villi over here, so this is intervillus space. This is also some villus, which is bigger one here on the right, and this intervillus space is filled with mother's blood. So sometimes we can see erythrocytes over here. Can you see this nice little erythrocyte over here and here? So these erythrocytes belong to the mother. And these erythrocytes belong to the fetus. So the diffusion of nutrients takes place in here, in some way here. So gradually we reach the development of the placenta on the side of the chorion frondosum, and this image over here combines a timeline, and at the same time as the chorion grows, as the individual villi will change. So here we have days. First we have some primary villi over here, just made of cytotrophoblast and syncytial trophoblast. Here we have secondary villus. Here we have days, so you can see in second week, after second week, we develop tertiary villi over here, because we now have vessels inside. And what is interesting is that cytotrophoblast, this layer over here, cytotrophoblast will really gradually go or grows into the decidua over here. Here we have a decidua, so this is decidua. You can see the cytotrophoblast, which is this violet color over here. This is the cytotrophoblast, and I'm sorry, this is the I'm confused. This is the syncytial trophoblast over here. Syncytial trophoblast, it's written right here. And cytotrophoblast behaves interestingly because it starts to grow, and it grows to the decidua over here, to the other side. Yeah, first we have the trophoblast cells on the villi here, and they will grow to the other side, to the decidua, and they form a plate, this plate over here, and this plate is called a cytotrophablastic shell. Cytotrophablastic shell over here, the layer of cytotrophoblast here. And this, why I'm mentioning this is very important, because we have this shell at the point where it comes into contact in the mother's blood vessels. Yeah, so here in the decidua, the basal decidua, we have mother's vessels, and this cytotrophablastic shell will come in contact with this mother's vessels, and the cytotrophoblast begins to penetrate and replace some endothelial cells. So the so-called hybrid vessels are formed where the endothelial cells of the mother and cells of the embryo, the cytotrophoblast, are combined together. So cells, really imagine that cytotrophoblast will mix with endothelial cells of the mother. So two different genomes will combine. And this is a very complex system of immunotolerance that has to develop here. Yeah, because mother's immunity here is like, whoa, we have cells with another genome here, what's happening? So there needs to be developed some kind of immunotolerance here in mother's vessels, the mother's blood, really, to accept the embryo. This is some general view, probably fifth week of development. The embryo, as you can see, is not growing much. It's very small right here. It's not growing much. And during this embryonic development, we reach about 3 centimeters. Yeah, so within 8 weeks of development, we reach 3 centimeters, and weight is only about 2 grams. Yeah, so during embryonic development, 3 centimeters, 2 grams of weight. So the growth of the embryo itself is very minimal. You can see it in this picture. It's a very small embryo here. Only in the fetal period, embryo really starts to grow in the fetal period right here. And we can see right now in this picture, we can see a clear difference between the chorion frondosome, which is the part that is over here, the chorion frondosome. We can see nice villi over here, which are branching. Yeah, so this is the basal decidua here in the uterus, basal decidua here. And we can see the stagnation of other chorion on the opposite side, the smooth chorion here. So this chorion here is very, very small. There are almost no villi here. This is normal, it's chorion leva. So in chorion frondosome, the villi are mostly in tertiary, and in chorion leva, the villi are mostly secondary here. There is some kind of stagnation. So this is just a general view right here, and you can see maternal vessels that go to the lacunae here, to the intervillous space, and there is a blood here, and also maternal vessels. Not only arteries are going there, but also veins are there, because somehow we need to transfer blood back to the system of the mother. Yeah, so blood is going this way, and after deoxygenation it's going another way through vessels, the veins, over back to the mother. This is an image with an embryo injection and some staining. So what we can see very nice vascular system, these lines over here, which is very rich in chorion frondosome over here. So this is a nice little embryo right here, as you can see this picture, and there's are the vessels in a chorion frondosome over here. So this is the chorion frondosome, and we can see that it is a villus, so you can see the villi over here. It's branching. So during the late embryonic and especially fetal period, the embryo finally begins to grow, because we need the bigger embryo, really, the fetus right here. And its envelopes began to merge, because now you can see there are many envelopes here. There are many layers that are around embryo, and at the end we have only one envelope, one amniotic cavity here. So they need to merge, and as the fetus grows, they will merge. So what is really happening? Originally, I'm going back to this picture. As you can see, there is an amnion or amniotic cavity, the whole cavity around the whole embryo here, and it's very small. Small cavity surrounding the entire embryo here. The chorionic cavity, right here, the chorionic cavity is very big. It's very large here in the 5th week. But what is going to happen? At 8th week, however, there is no such big difference as before. Here we have big amniotic cavity around embryo, amniotic cavity, and chorionic cavity isn't that big. It's almost the same as amniotic cavity. So the embryo is bigger and these cavities are the same, almost the same. And the amniotic cavity expands, and really what's this layer over here, the amnion layer, is a layer formed by special cells called amnioblasts, because it's amniotic cavity, so there are cells called amnioblasts, and also somatopleura, so there are cells like the blast, and somatopleura approaches the chorion. So it's going to near to the chorion over here, going close to it, and these layers fuses together, because also the chorion here contains somatopleura, it contains mesoderm. So two mesoderms approach together and they merge, and they merge and they are leaving only one cavity there, and that is amniotic cavity. Just try to imagine what layers do we have on the amnion, what layers we have on the chorion itself, and when it gets together it just fuses and creates only one amniotic cavity here. What's happening around 12th week? We see that those layers are already fused, so we have one amniotic cavity. It's sometimes called amniochorion because it's really amnion and chorion together. But the cavity is just amniotic cavity. Just call it like that. And really the chorionic cavity actually disappeared. So it just disappeared. So when there going to be a question, what happens to chorionic cavity? The right answer is it disappeared. It was fused. Really, the two layers fused together, and there is just only one cavity here. Here you can see also another cavity over here, and that is uterine cavity. So of course we have some space also in uterus, so there is some cavity here. And also uterine cavity will almost disappear because the embryo that grows in the lining of the uterus, the embryo grows in the lining of uterus or the mucosa here, that is covered by decidua capsularis or the capsular decidua that we described earlier. It begins to arch and approaches the lining of the uterus all around. So also uterus from one side will fuse with the mucosa of the uterus on the other side, and the uterine cavity will also disappear, And the decidua that is around, I didn't mention that. I just mentioned decidua capsularis, the basal decidua, and the marginal decidua that is between basal and capsular decidua. But we have something also called decidua parietalis or parietal decidua that is all around the uterus. So capsular decidua will fuse with parietal decidua, and uterine cavity will also disappear. So try to memorize and imagine these two pictures together. It's sometimes complicated to imagine. Yeah, there is only a small space that is left over here, small space over here, and this is the very small uterine cavity right here, the remnant of uterine cavity. Okay, so as a summarization, the original three cavities disappear. Yeah, only the amniotic cavity and only a tiny cavity of uterus remained. Yeah, so now we can see another structure over here, umbilical cord. And so the embryo is connected to placenta via umbilical cord, and really the placenta is made of chorion frondosum right here and decidua basalis or basal decidua here. So placenta has two parts really. One part that belongs to fetus and one part, the basal decidua, that belongs to the mother. So we will talk about umbilical cord more, but first let's have a look at the video about bending of the embryo, the bending right here, or the folding. Yeah, folding. We have folding in a transverse plane and also in the sagittal plane, and it's a 4th week event, really, the folding of the embryo. It begins in the 4th week. It's a change from a flat to a tubular C-shaped embryo. So what we can see right now, we see a trilaminar embryo over here, trilaminar embryo, and we have amniotic cavity over here. So this blue one is amniotic cavity on the top and yolk sac at the bottom. Cranially, there is a cardiogenic zone is created in front, where we can already see the pericardial cavity here. This space here, this is the pericardial cavity. So far, however, everything is quite straight. Yeah, nothing is folding right now. So this is the 3rd week here. So first let's have a look at folding in the transverse section. So we have 3 layers because this is a trilaminar disc, trilaminar embryo. So this is the section, and now we have a transverse section over here. In the middle, we can see the notochord right here, this green one. Here we have a neural groove over here. We can see the 3 layers, the ectoderm, the mesoderm that is red, and also endoderm, which is yellow right here, so 2 layers. And now let's have a look at the folding in the transverse plane right here. Yeah, so nice notochord and stuff. So we can see nice neural groove right here. And the important thing is going to happen right here. We have these lateral structures over here, and these structures are from amniotic cavity. These are called folds, amniotic folds right here. Yeah, so this is the transverse section, so that's why they are called lateral folds. Yeah, folds of amniotic cavity right here. They compress the yolk sac, as you saw here. They compress the yolk sac, so yolk sac before was bigger and it's going to be compressed because of the folding of the embryo, and there is only a small thin duct that connects the yolk sac and the developing gastrointestinal tract over here is connected to yolk sac with a duct, and this duct is called omphaloenteric duct. Omphaloenteric duct. Also, because of the folding, we created another cavity here that is called intraembryonic coelom, or the body cavity, because of the transverse folding. Yeah, and slowly it's going to close itself because of the folding. Here we can see it's still open into the chorionic cavity, which is around here. So around the embryo we have chorionic cavity, which is quite big, and inside in the embryo we have intraembryonic coelom, and it's communicating with chorionic cavity over here. The duct, the omphaloenteric duct, which connects the embryo to the yolk sac, will close completely, but not right here in this stage in the 4th week. It will close in 11th week. So it will close further in time, not now. But we can see in this picture that the folds are going very close together right here. And there is still this ductus remaining right here. But when we make a section more cranially to the cardiogenic zone, they are going to show us the folding once again of the embryo. So we need to wait a little bit. And it's more cranially to the omphaloenteric duct, so the folds will fuse together, and the intraembryonic coelom, this body cavity, will close. Yeah, but remember, there is still some way behind this section. There is still some connection. There is still omphaloenteric duct, which is connecting yolk sac to the gastrointestinal tract. So there is always some kind of connection between the chorionic cavity and intraembryonic cavity. Yeah, I hope you do understand. So let's go to the sagittal view, probably the last view, folding in sagittal plane. So what we can see right here, 3rd week, and we are starting 4th week because we start to fold. So this is the chorionic cavity around. There is an amniotic cavity. Here we can see really nice neural folds and stalk, connecting stalk, which gives rise to umbilical cord. And now what's happening, we have also these lateral folds, as we saw before, but now we have cranial fold right here and caudal fold right here. So together we have 4 folds, really. 2 lateral and 1 cranial fold and 1 caudal fold. But what is happening, these 2 folds in the sagittal plane don't fuse together as lateral folds. Yeah, they are just close together because of the folding, but they don't fuse together. Once again, here we have yolk sac and the developing omphaloenteric duct, which is connecting yolk sac to the primitive gastrointestinal tract over here. Yeah, so let's go back to the presentation. You can find this video also on Moodle online. And now I'm going to explain what's really happening. This is also the sagittal view here. And here we can see this structure over here. Here we can see the cranial fold, amniotic fold. So here we have amniotic cavity on the top, and there is the caudal fold, which is a little bit smaller because everything is bigger cranially. Yeah. So the embryo is growing in cranial caudal formation. So it's growing more and faster on cranial parts than in caudal parts. Yeah. So this growth or this fold is bigger. Okay. In this case, the omphaloenteric duct, which is over here, this structure over here, is very wide. But the folding of the embryo will narrow this omphaloenteric duct. Remember that. It really, because of the folding, this omphaloenteric duct is very close to this connecting sulc over here. It's very close to connecting sulc. So they get together, and you can see there is a protrusion from the primitive yolk sac. There was a protrusion called allantois. So you can describe allantois as a protrusion of a yolk sac that is protruding to the connecting stalk over here. The umbilical cord, really. So once again, if you want to see, here we have, oh sorry, I wanted to change colors. Here we have primitive gut over here, which is dividing into many parts. We're going to learn about that later. So this is the primitive gut, which is connected by a ductus omphaloentericus with yolk sac over here. There, everything is narrowing. They are together. And finally, we have such situation. We have a ductus, which at the end extends into yolk sac and connecting stalk. And among these two structures, there are still remnants of space. Yeah, there is still space between these two structures, and this space is connected to chorionic cavity. So there is still some connection with chorionic cavity. Sometimes this space between the yolk sac and the duct and connecting stalk is called umbilical cord coelom. Yeah, but it's really chorionic cavity, and it's called sometimes umbilical cord coelom. Yeah, this is the place where the chorionic cavity is preserved for the longest time, because you now know that chorionic cavity will disappear and the amniotic cavity will get bigger and bigger, but this is the place where it stays for the longest time. So we will see in the moment these two structures, the omphaloenteric duct and the umbilical cord, the connecting stalk, will be connected together. We can see it here in this situation. Once again, we have amniotic cavity around embryo, and here we have chorionic cavity, but amniotic cavity will get bigger and bigger. They are like pushing together these two structures that we saw before, and they will eventually fuse together. Yeah, we can see it in the next picture, the 10th week. These structures will fuse together. Yeah. Maybe I'm going back because these structures together, the omphaloenteric duct and the connecting stalk, are together creating the umbilical ring. Yeah, an umbilical ring right here. And we can see it also like this right here, umbilical ring around these two structures. So here we can see that we have also blood vessels around this structure. So we have some vessels around yolk sac, the duct, and we have also some vessels in a connecting stalk, of course. And there will be some reduction in these vessels, and the most noticeable reduction of vessels in the umbilical cord, because first we had two pairs. We had two umbilical arteries and two veins, but at the end one vein will disappear. So we have just two arteries and one umbilical vein over here. And also for the vitelline vessels, they will disappear mostly, like all of these vitelline cells will probably disappear in some time. Yeah, we're going to talk about it more in the cardiovascular system. Umbilical vessels are really essential for supplying the fetus with nutrients, as you probably know, and on the oxygen from placenta. Oxygenated blood comes into the fetus through one umbilical vein, and the oxygenated blood enters the placenta through two arteries. So here we have deoxygenated blood, and in the vein we have oxygenated blood, and everything is fused together, yeah, right here. And in some case we will have an intestinal loops here. So first we have vitelline duct over here, that's just omphaloenteric duct in another name, and after that we're going to have intestines over there, and that's pretty normal. I'm going to talk about it. And I talked about that before, that the omphaloenteric duct will disappear in some time, and also yolk sac will disappear in some time. And when it's going to happen is that you already know that omphaloenteric duct is connecting yolk sac and intestines right here. And what is going to happen in like 7th week is that intestines will go out from the abdominal cavity right here in a way of physiological herniation right here. And the loop, the intestinal loop, will come out, the one that is connected to the yolk sac right here. It will come out because intestines are very big for this small embryo. Yeah, they are very big, but in some time they will need to go back to the abdominals, because we are not just walking around with our intestines outside of our belly button. So they need to go back to the embryo, and when they are going back, when there is enough space for intestines, the herniation will disappear, and in that case also omphaloenteric duct and yolk sac will disappear eventually. Yeah, so just remember right now, in the process of reposition of herniation, the physiological herniation that's happening right now, the duct and also yolk sac will disappear, and that's about in 11th week. So what's really happening? First, we have this kind of situation when we have a vitelline duct of omphaloenteric duct with vitelline vessels around, and we have allantois, which is a protrusion of yolk sac here, right here, and two arteries and one umbilical vein or umbilical vessels, but they're going to disappear. You can see that the vessels, the vitelline vessels and also omphaloenteric duct will disappear eventually, and what stays here in the mesoderm, extraembryonic mesoderm, is the allantois and the vein and two arteries, and they're getting bigger and bigger. So now in the umbilical cord we have two normal arteries, one big vein, and sometimes we can see in the center the remnants of allantois right here. We can see it in some slides also, I will show you probably. And all around we have more differentiated mesoderm that is called Wharton's jelly. You already know that, Wharton's jelly. Yeah, or mucous connective tissue right here. in the umbilical cord. Umbilical cord is covered by amniotic epithelium on the surface. So let's have a look on the slide, the histological slide. This is the vein. These are the two, oops, I'm sorry, but you can see it. These are the two arteries over here. Yeah. So this looks like really some scared face over here. And this is a remnant of allantois. So we can see allantois here, and sometimes you will be able to see it also during practical trainings. There is some epithelium on the surface, and this is the amniotic epithelium here. These are the amnioblasts on the surface over here. Okay, and this is Wharton's jelly all around the umbilical cord right here. We can see it also right here. There is an embryo. Here is a very, very, very thin omphaloenteric duct with yolk sac over here, and this omphaloenteric duct is connected with umbilicals. Also, this is umbilical cord right here, this thick structure over here, and it's going together with this ductus omphaloentericus. And here in the next picture, it's very interesting, we can see the herniation really to the umbilicals. Yeah, the herniation of intestines over here, so-called physiological hernia, because it's very normal and physiological. And when this herniation will go back, we will lose the yolk sac and also the omphaloenteric duct. So, the placenta develops from the chorion frondosum and the basal decidua, as we said before. Yeah, chorion consists of extra embryonic mesoderm, which also forms jelly in the umbilical cord. Yeah, and chorion also consists of trophoblast. So trophoblast and mesoderm make up the chorion. Chorion is made of a villi. Yeah, between villi we have intervillous space, which is filled with maternal blood. Intervillous space between villi. Villi are covered with trophoblast, of course. Here we can see the known cytotrophoblastic shell over here, which is very important for the formation of hybrid vessels. Here we have really individual lacunes over here. They merge into one space. And this is the intervillous space, and we can talk about fully developed placenta around the 15th week of development. Yeah, so around 15th week we have fully developed placenta. So maybe remember this time right here, this week. So that's when we talk about the developing placenta. The chorion frondosum differentiates into a chorionic plate. Right here, we have the chorionic plate also. What I wanted to say, I forgot. And yeah, okay. These are the chorionic plate, and here we have umbilical vessels inside. You should, like, can you see that? There is a connecting stalk over here, the umbilical cord, and here are the umbilical vessels in the chorionic plate also, which are branching, and they create these capillaries in the villi that are here. So these villi contain fetal blood, and maternal blood is here in the intervillous space. So here we can see it once again, the developing of the placenta over here, and what is happening really. There we have a chorionic plate and vessels. It's not so different as this picture over here. I think it's probably the same, the developed placenta. And we have some basal decidua here and the chorion, which is making up the placenta. You can see here the decidua is protruding into septa. We call this septa decidual septum, and these septae are dividing placenta into parts, and these parts are called cotyledons. So this part from one septum to another septum, like this, I will call this part cotyledon, right here. You can see the fetal part right here. The fetal part is covered with amnion, the amnion. We can also see it in this picture. Here is an umbilical cord, and you can see this part over here. It's shining, it's covered with amnion, yeah? And this is part that is attached to the wall of uterus in the mother, and this part isn't covered with anything. And here we have these parts called cotyledons, so you can see these grooves between cotyledons, and this is because of the decidual septae that are going deep into the placenta. We are also able to recognize the decidual septae in histological specimen also. So these are the general things you should know about placenta: the diameter, the thickness, you can read it right here, the weight, yeah, but remember there is like half a liter of blood inside the placenta, so together is like 1 kilogram of placenta. It's very, very heavy structure, really. So it's discoid, that's why it's called discoidea, yeah. And also, so it's like in disc shape. It's having cotyledons, which I described. These are these bulges right here, cotyledons. That's why it's called cotyledonata, and it's a difficult name. And it's also hemocorionic, hemocorialis, and this means that the mother's blood directly washes the chorion. Yeah, remember intervillous space? It's in contact. The mother's blood is in contact with the chorion. So it's needs to be the immunocompetence here and the immunotolerance here needs to be developed very well. So here we have a nice picture from scanning microscope right here. So just for your look right here, and here we have some villi. You can see there are these 3 villi, the branches of another big villus that is somewhere deep inside. So these are the villi over here, but also there are these big villi which are connecting basal plate with chorionic plate over here, the decidua with the chorionic plate, and these villi, I cannot see them directly here, but these are called anchoring villi that are connecting basal plate and chorionic plate together. And we can see it right here. Now this is another picture from placenta, and we can see one anchoring villus over here, which is branching into smaller terminal branches right here, the terminal villi, which are of course tertiary villi because we are in fully developed placenta right here. And yeah, that's this anchoring villus is connecting the chorionic plate and the basal plate together. Okay, this is a picture from electron microscope here. We can see this is the villus. This is a tertiary villus right here, the whole picture. We can see it contains capillaries right here with erythrocytes in here. So you probably know these things right here. These are the erythrocytes. These erythrocytes, these vessels belong to fetus, of course. We cannot see any maternal cells. Probably there is intervillous space around here, so there should be also some erythrocyte. It's not really a biconcave shape, but whatever. There are some erythrocytes. So, and here is the placenta barrier right here. So how would you describe placental barrier? Is that it's made of syncytium, Yeah, syncytia trophoblast. I need to write like syncytia trophoblast, because we can see syncytia trophoblast here. There are the cells all around covering the villus, and syncytia trophoblast has its own basal lamina. Basal lamina of syncytia trophoblast, and then we have basal lamina once again, but this time this basal lamina belongs to endothelial cells right here. I was writing it too low. Endothelial cells. Endothelial cells are cells that are covering the capillaries, that are inside the walls of vessels. Yeah, so this is really the placental barrier right here. The nutrients and oxygen need to go through syncytia trophoblast and basal laminas here that are really fused together. They are fused together, and endothelial cells to get through. So here we have a nice picture of the decidual septum. We know it's decidual septum because it's more eosinophilic. It contains decidual cells. You will learn about it more next semester. So this whole structure is decidual septum, which is dividing the placenta into parts called cotyledones. Yeah, but remember, the decidual septum is not connecting chorionic plate with basal plate. Yeah, it's just protruding from the basal decidua. It's going to the way of chorionic plate, but it's not connecting these two structures. On the other hand, anchoring villi are really connecting two plates. Yeah, the maternal part with the fetal part. There is this connection, but the decidual septum is just heading to the chorionic plate, but really not touching it. These are the villi, the 3 villi that are here, many sections of them. So now let's jump on the twins. The last important thing I want to talk about are twins, and we distinguish dizygotic twins right here and monozygotic twins right here. The dizygotic twins, you have 2 eggs that are fertilized by 2 sperms. So we have 2 embryos developing together in one uterus, and there are only 2 situations that can happen. So they can be implanted, these 2 blastocysts that were created can be implanted close together, like in this case, or they can be implanted separately and develop completely separately. And these 2 embryos do not compete with each other, so this is a very good situation. Yeah, everybody has its own place to be developed, and everything is just fine. But when they are close together and when they fuse together a little bit like this, probably they have fused placentas also, and this is the problem. This is very bad because embryonic nutrition is not always ensured in this case, because fused placentas can lead to malnutrition of one embryo or even to death of one embryo. Yeah, over here, because there are more nutritions that are going to one side and less nutritions are going to another side. It's not always as equal as is in this case in the picture. Yeah, sometimes there is just a little part of placenta that is here and more placenta is on the other side, so it's not very good situation, but they can develop normally. Yeah, they can develop normally, but sometimes there is malnutrition to one embryo, and that might be a problem. Then the next variant, the second variant, is monozygotic twins. Yeah, monozygotic twins. So it's about fertilization of one egg with one sperm. And it really depends on what stage the separation takes place. This can happen very soon, the separation before the blastocyst stage. Then we have two blastocysts. Yeah, and the situation is probably the same as for dizygotic twins. So they can be implanted close together, or they can have fused placentas and stuff, but we have two blastocysts. Splitting or division can also occur in the blastocyst 2 or 3 days later, where the embryoblast divides into two nodules. So we have two embryoblasts over here. So it's implanted. Normally, it's implanted. And each embryoblast forms its own amniotic cavity. But the chorion is only one for both embryos, for sure. There is no other option, actually. There is no way there will be separated placentas over here. They have the same chorion, the trophoblast, and really they have separated amniotic cavity, but they have one chorionic cavity, so they have only one placenta for both of them. This can also be a very dangerous state for the embryos here, because of the malnutrition that can occur. Now we have a third option right here. So really what can happen also is that the splitting will happen after the formation of embryoblast, and you can see that it's not even split. Yeah, so there is no splitting of embryoblast. Actually, I said it wrong. There is no splitting of embryoblast, and the two embryos develop like this. They have same amniotic cavity. So here we have one embryo, and here is the second one embryo. So they have the same yolk sac. They have the same amniotic cavity. And what's really happening is that you develop two embryos in one amniotic cavity here. Yeah, so this is a very dangerous situation because they're competing with each other and they are very close together, and this is very dangerous. Really, the most dangerous probably stage that can, or the thing that can happen. So just remember the difference. Yeah, and what can happen when the blastocyst will split, and embryoblast splitting right here. We can see that two embryos, two amniotic cavities, but one common placenta. And when it don't split at all, and we have two embryos in one amniotic cavity, like this. So this is the last part of my presentation, and this will be very quick. You may already know the Czech embryologist, Professor Jirásek. The professor has been involved in the systematization of embryos all his life, and he has created his own system. It has a certain advantage, this system, namely that it also includes higher stages of development, unlike the second classification, which I'm going to show you, the Carnegie classification. So far we have been able to describe the embryo to stage 5. We will describe it now from stage 6. I will show you some pictures. It's really not necessary for you to know all details, but let's have a look on some pictures, and yeah, so let's start. So from stage 6 we have a tubular embryo. Then we have stage 7, a C-shaped embryo. Yeah, the tubular embryo is called because of the closing of neural tube. And then we have C-shaped embryo when we have a formation of limbs right here. And there is this late embryonic stage where limbs are fully differentiated, including fingers and toes and the closing eye fissures. Then we have a fetal stages and perinatal stages. So these are the weeks till 8 week. This is embryogenic stage. Yeah, embryogenic stages. And this is the fetal stage right after 9 week of development. And let's have a look. This is another classification. These are the Carnegie stages. And this system is based on examination of typical embryos of certain age categories. But there are only 23 stages till 60 days, so it's not till birth actually. In the following pictures we will use both stages. You don't need to know those stages, yeah, it's just for you to visualize stuff. So stage 6 is further divided into another 3 substages, as you can see right here. The tubular embryo, because we have a closing neural tube, is formed, and also it's formed because of the expansion of intra-embryonic structures like somites, yeah, and this neural tube, as I mentioned before, and also the vascular system, heart tube, and heart loop are formed. This 6th stage lasts 10 days. It actually covers the 4th and part of 5th week of development. The embryo is elongated and the entire length of the embryo is measured up to 3 millimeters. 3 millimeters, so it's very small, and it's not very well supplied by the vascular system, as you know. And probably we can see some pictures. You can read about things that are going on in each stage. We can see how it looks like. There is this, we can form this posterior neuropore and anterior neuropore. We're going to learn about it in development of neural system right here, but you already know something about somite here, somite. And here we have a nice neural tube that is closing right here. So this is the 6th stage right here. And this is how it looks like on the picture. So have a nice look. It's very pretty. Yeah. Here we have a bigger anterior part, the smaller caudal part or posterior part. Yeah. This is anterior part right here. So this is a picture of developing embryo right here. Somites, as you probably know, 3 somites are created every day from the 23rd day, and this makes it possible to recognize the day of the embryo. Yeah, the 3 somites are created each day. So this is a very nice picture once again. Here we have nice somites. So these are the somites over here. They are perfectly visible because they push ectoderm about them. Yeah. So here we can hear it once again, and also the section. Here we can see the cross section. So very nice pictures, and the somite once again. And wow, these pictures show us probably the dorsal flexion, little dorsal flexion of the embryo. The dorsal lordosis occurs here. The neural tube that is here actually causes this dorsal lordosis here, but it's not so important for you. We can get to the next stages over here, the 7th stage of development. 7th stage overlaps the 5th, the 6th, and 7th weeks, really. And we have many stages in this one stage. So you can see development of many things over here. And the embryo grows. We measure its length from the most cranial point to the most caudal point, and it grows up to 24 millimeters. Yeah, so it's quite big. It's bigger than 3 millimeters before. It weighs up to 2 grams, and it's very important for Irasek's division. He focused on developing of limbs and face. Yeah, so based on development of limbs, we have many stages here in 7th stage. So Irasek divides the stages into another stages with, we can see that we have here the buds, arm and leg buds. Then we have bi-segmented arm buds, yeah, and then after that we have 3 segments of like the arms and legs. The first picture where there are limb buds is right here, because we can see the buds, limb buds over here. This is one from one side, and these are the developing upper limbs over here. They are formed only at the level of the 5th, 6th and 7th somites. We also see larger limb buds, but we still only have upper and a single segment right here. So these are actually the upper limb. You can see they are quite low, but this is where they developed. And first we have upper limbs, and then we have lower limbs, but they are not here. They are not in this picture. We can probably see them in the next pictures. Yeah, we can see them in this picture. So here we have, you can see they are bigger over here. Yeah, but we still have, they are larger right here, but we still have only upper right here, and single segment is only here, the upper buds. Next picture shows the upper and lower buds over here. So now we have upper and lower buds. At the top we can see maybe some thickening and transition to the 2 segment, but not really right here. We cannot see it clearly, but they are going to be transitioned right after. Yeah, so once again, these are the upper, these are the lower buds over here. Here we can also see, we can see quite well 2 segments. Yeah, 2 segments. So bi-segmented leg buds and also bi-segmented arm buds. And we can very clearly see the eye, developing eye of the embryo. So it's very nice. And this picture, there is special labeling here. We labeled chondroitin sulfate, so we can see the cartilage over here, and we can see the limbs have 3 segments over here, 3 segments. And also we can see developing fingers over here. It's very nice. This is the eye, and also face is developing, so it looks like more and more like human, normal human. This is the lower leg, which is a little bit smaller, but never mind, it will grow also. It's normal. And here we have next the picture with embryo. This is the embryo with fingers also. We have fingers and toe tubercles right here. So really this is an embryo at the end of stage C. Yeah, so the 5th day, 45 to 50 days, it looks like this. So this is the late embryonic stage, and the primary palate is closed. We have open eye fissures, yeah, because fusion of eyelids is regarded as the end of embryonic period. So at the end we still have open eye fissures, and when we move to fetal stage, which is from 9th week, the fissures are closed. The eye fissures are closed. And we have this measurement. The embryo is big, about 3 centimeters big, and we measure the embryo with ultrasound, and it's called crown rump length. Crown, it's like the top of the head, and the rump is the bottom of buttocks. Yeah, so bottom of buttocks, and it's crown rump length. You can find it on the internet, and for each stage of development there is some kind of standardized measure, and based on that you will know if the embryo is nourished well, or if there is some kind of retardation or something bad going on. So crown rump length, you should know that. So first we have embryo with short eyelids, and then we have embryo with fusing eyelids. So probably you should know that eyelids are fused in fetal stage. So we are at the end of the embryonic period. Here the eyes are still open. Right here you can see that they're still open. They're not covered by lids, and that's normal because lids are just growing and they fuse together. But you can see that the embryo has all its fingers separated right here, also on the arms and legs, and the face is slowly changing to a human face right here. So it will be a human. Yes. And so these are already fully developed embryos right here, which I'm showing you. These are lids that are fusing together, they are closing together. Nice. And here we can see fused eyelids together, so eyes are closed here. And once again, the fusion of eyelids is regarded as the end of embryonic period, right here. And now we have fetal period. So, the first and the second trimester fetuses, really, the fetal period, right here. So, what can I tell you about fetal period? These are the calculation according to gestational age. So as you probably remember from previous presentation of development, there is some anatomical age of the embryo or the fetus, and gestational age of the fetus. And for example, it's counted like this. For example, 11 gestational week is 9 weeks in after fertilization. Yeah, so it's 9 weeks after fertilization actually, but in gestational weeks it's +2. There are more. Yeah, so you should remember like this. During in gynecology they will be using gestational age of embryo, so you should know it's like +2 to normal anatomical age of the embryo. So this is these are the weeks after the fertilization. So the fetal period lasts until the 28th weeks of gestational, or 26 weeks of anatomical weeks. Or I should call it. Yeah, you remember. So the fetal period lasts till 28 gestational weeks. Okay, so and until that the embryo is like 50 times bigger. It's heavy. It's 1 kilogram. The weight is 1 kilogram. Yeah, so You should know that fetal period lasts from 9 to 26 anatomical weeks that we are normally counting here in histology. So, and this is the fetus in first trimester. Very nice picture right here. You can see the head is very large, and it's going to get smaller, or the body is going to get bigger. So in this picture you can see it's more proportional in the second trimester fetus. Here is nice umbilical cord over here. And next we have perinatal period right here. So perinatal period is after 26 weeks till birth, yeah. So, and we can measure weight of embryo, and based on that we divide newborns into immature ones that are very small, that are like 750 grams small, and very low birth weight, premature newborns, normal newborns, which are this big, and macrosomia, which is like hypertrophic newborns that are over 4.2 kilograms. So we divide them based on weight. And at the hospital, it's when the child is born, the newborn, we don't measure the crown rump length anymore, but parietal length is measured, which is normally about 50 centimeters. So normal newborn should be about 50 centimeters long and about 3 kilograms of height. And this is how it all ends. So this second part of the presentation wasn't so important, but I showed you some nice pictures. I hope you like them. You don't have to know the classification of Professor Jirasek, but the gynecologists probably do know this classification, but it won't be on test or during oral exam, so don't worry. But you should know the amniotic cavity, the chorionic cavity, the development of trophoblast and stuff I talked about in the first hour of presentation. Okay, so I hope you enjoyed this presentation, and goodbye.